Welcome to NIH 101, Peer Review and Priority Setting. I'm Howard Kurtzman, the Acting Executive Director for Science at the American Psychological Association. My job today is to provide a bit of background on NIH and to introduce our distinguished speaker, Dr. Keith Yamamoto. But before I do that, I want to acknowledge the sponsors of the briefing, the Coalition to Promote Research, the Coalition for Life Sciences, the Ad Hoc Group for Medical Research, and the other organizations listed on the slides on the wall here. I'd also like to thank the Congressional Biomedical Research Caucus for its support of NIH and for its help in securing our room and promoting today's briefing. We particularly thank the caucus co-chairs, Representatives Steve Cohen, Jackie Spear, and Steve Stivers. The National Institutes of Health is often called the crown jewel of biomedical and behavioral research. From modest beginnings in the 1930s, the NIH has grown into a $30.3 billion enterprise made up of 27 institutes and centers. NIH funds research not only at its own laboratories, but in all 50 states and the District of Columbia. In fact, more than 80% of NIH's funding is awarded through competitive grants and contracts to institutions throughout the U.S. and in some cases in other countries. Currently, NIH funds almost 50,000 competitive grants to more than 300,000 researchers at more than 2,500 universities, medical schools, and other research institutions. About 10% of the NIH's budget supports projects conducted by the nearly 6,000 scientists in its own laboratories, most of which are on the NIH campus in Bethesda, Maryland. In fiscal year 2012, NIH-funded research supported an estimated 402,000 jobs across the U.S. In addition to directly supporting research, NIH funding helps to generate spin-off economic growth, ranging from suppliers of scientific equipment and information technology, to biotech and pharmaceutical firms, to businesses offering food and lodging. A report from United for Medical Research estimates that research supported by NIH generated $62 billion in new economic activity in 2011, more than double the taxpayers' investment in the agency. Of course, NIH's work to improve health and save lives is the reason that the agency has enjoyed bipartisan support in Congress. The life expectancy of Americans has increased steadily over the last century, thanks in part to scientific advances supported by NIH. NIH-funded research has led to the development of vaccines to protect against such diseases as influenza, meningitis, and cervical cancer. Death rates from cardiovascular disease have declined by more than 70% since 1963, with more than half of that decline coming in the last 20 years. That decline is the result of improved treatments for cardiovascular disease developed on the basis of NIH-funded research. It also reflects NIH's investment in research on how to help people quit smoking and prevent people from smoking in the first place. These examples illustrate that progress is made by NIH's support of many different types of research addressing a wide range of conditions. NIH is organized and managed to ensure that it funds the highest quality research in areas of greatest scientific opportunity for improving health. It is a large and complex task. So it should not surprise us that NIH does not look like other agencies and that its system of peer review and priority setting might require some explanation. Keith Yamamoto's experience makes him the perfect guide to how NIH works and how it interacts with the scientific community and the public. Keith is at the University of California, San Francisco, where he is Vice Chancellor for Research, Executive Vice Dean of the School of Medicine, and Professor of Cellular and Molecular Pharmacology. He is a highly regarded scientist whose work focuses on communication and regulation within cells, including the mechanisms by which hormones affect the functioning of cells. He has led or served on numerous national committees concerned with scientific and public policy, public understanding and support of biological research, and science education. He chairs the Coalition for the Life Sciences. He serves on the Council of the Institute of Medicine and on the Advisory Committee of the National Academy of Sciences, Division of Earth and Life Sciences. Keith has chaired or served on various committees that oversee the process of peer review at NIH, including as a member of the Advisory Council of NIH's Center for Scientific Review. He is also a member of the advisory board for the Lawrence Berkeley National Laboratory and the board of directors of the advocacy group Research America. Among his honors, he has been elected to the National Academy of Sciences, the Institute of Medicine, the American Academy of Arts and Sciences, and the American Academy of Microbiology, and he is a fellow of the American Association for the Advancement of Science. I am pleased to introduce Keith Yamamoto.
Thank you. It's a pleasure to be here. For a very nice introduction. Sounded ominously like an obituary, but let's just see if we can get through that and get on to this thing. This is a very interesting time to talk about this issue of the NIH because of the tremendous opportunities that have been generated by previous NIH-funded research and new policies that are moving through the agencies, things that have been set as priorities by the administration, and tremendous opportunities on the research side. So it's really a nice time to be talking about this. And we'll talk about both this notion of peer review that Howard mentioned and priority setting. To do that, I want to back up and talk a little bit first about biology. Introduce you to biology and what the way that biology works that flavors the way that biological research is done, which in turn flavors the way that the funding that the NIH works in the funding realm. So I'm going to start with this. It's obvious, but bears a little bit of drilling down on that biological systems are complicated. And if you look at some of the tasks that biological systems carry out, even at this kind of 30,000-foot level, you can see that things are not that simple. Your genome is made up of tens of thousands of genes, and what makes your cells different is not that they contain different genes, but some genes are turned on and some genes are turned off. So turning genes on and off is a complicated process. Obviously, going from fertilized egg through development to an adult is complicated. Deciding whether a cell is going to build up or break down fat, storing energy, or threatening obesity, and so forth. Going from a non-dividing cell to a dividing cell, obviously important during development, not so good during cancer. Neurons are born, but to really function, they've got to go to specific places in the body and make connections to muscles and other neurons, to sensory organs, and so forth. And making the right connections is a pretty tricky business. And going from a young organism to an old one, lifespan, aging, obviously important processes. So all of these things you can recognize are complicated, and the processes are not controlled by simple switches. And in fact, they're not even controlled by long, multi-step linear pathways. And that complicates things. So this hard wiring, if this is the way things worked, would render us unable to sense and respond to changes in time and environment. So we're not hardwired, right? And that's what makes these processes complicated and makes the idea of a simple switch or a linear pathway not really viable. And instead, what we have is this. So genes work together in networks that communicate with each other. These are proteins. This is one little network switch in the body looking at one function. And you can see that it's not quite exactly linear. And the genes are interacting and talking with each other. And the reason that they're doing that is that there are sensors that are receiving information from outside of the cell, either physiologically generated, like from the brain, or due to environmental exposure, that's telling the body about what the current state of affairs is. You're hungry, you're tired, you're old, you're young. you've been exposed to environmental toxins or not, and so forth. All of that information is processed through these networks to give rise to some final output. So now you can see that if you're looking at the final output, right, understanding what you could do to intercept something that's gone wrong in this pathway that's led to a disease or an increased incidence of disease is not going to be trivial. And when you find a defect of that sort, right, understanding whether that's the one that makes a difference or not is not going to be trivial. And when you find such a defect, most of the time, the effect on risk, which is what we all hear about, risk factors went up a little bit, right, is modest. And so it's not just a straight line, oh, we found a disease gene, we're done, right? Or we found a disease gene, we understand the mechanism of the disease, now we can make a drug that'll stop that process, right? There are processes, first of all, that you don't really want to completely stop. And secondly, just simply unraveling the plate of spaghetti to understand what's going on is non-trivial. So the result, then, is biology is complicated, and since all disease is an aberration of the normal biological processes, it means by necessity, disease is complicated. So the goal of NIH research, then, is to understand biology as the only pathway to being able to understand and treat and cure disease. So if you look at what the kinds of questions that the public comes up with when you open the Post every morning and see a front page story that a new disease gene has been discovered, you start with, what is a disease gene? And if I have one, why does it only change the risk, sometimes by tiny amounts, that I'll get the disease? So the headline says, disease risk for this, if you have a mutation in this gene, goes up 5-fold. You think, oh my God, I better figure out whether I've got this problem. You read it in the second paragraph of the story, you see the 5-fold is taking you from 0.01 to 0.05% risk. And you think, what the heck's going on here? Why don't we know more? Why does disease diagnosis so often seem almost like guesswork? How could gathering more information about me, or about other people, or about experimental organisms like mice or fruit flies make a difference in my health? I'm perfectly happy to be interested in the health of my grandchildren, but I'd also like to know what effect this would have on my health. Those are pretty legitimate questions, right? And they're points of confusion for the public when they read about this. And when you see this as the end point of NIH research, you think, well, is this really making a difference? How can we move this forward? So it turns out that complicated organisms like us have evolved from simpler ones, of course. And the way that evolution from simple to complicated goes is by adding, not by taking the way that a simple organism works and trashing those mechanisms and making up new ones, but instead by taking the existing mechanisms and adding layers of increasing complexity. So you keep the stuff that you had, right, and add more. So that says that since they evolved by addition of new layers of sensors of the environment, regulators that are switching genes on and off and so forth, onto those that already existed in simpler organisms. Therefore, we can unpack the complexity of complicated organisms by basic research, that is fundamental discovery research about how processes work. In both, by doing the research not only in complicated organisms that we really care about, people, but in simpler organisms that have been on the way and they've stripped away some of those layers that are now making for the confusion of that plate of spaghetti that we looked at a few minutes ago. So, sorry, there we go. And that works. to unpack the complexity by doing basic research in not only complicated organisms, but simple ones. So if you look at where the big breakthroughs have come in the discoveries of some of those complicated processes that I put up in the first slide, it's pretty illuminating. So the breakthroughs for gene regulation, switching genes on and off that we talked about, came in bacteria and the viruses that infect bacteria. The big breakthroughs in cell division came in baker's yeast and in African clawed toad. Cancer genes were found in a chicken virus. The main developmental pathways that were really the breakthrough of thinking about how development actually works to start from a fertilized egg and build up the complicated shapes and systems that we have came in fruit flies, sea urchins, and fish. Understanding aging and lifespan came in a little soil worm, a little bit too small to see, about a millimeter long, and in studies of pond scum, really pleasant. Learning and memory, a sea snail. Those targeted connections that neurons have to make in order to go to the right places and do the right things, chicken. So it works. It works to study fundamental pathways in places where you can make progress. They're simple enough, you can do the experiments, and they've stripped away those extra layers of complexity. So we're not done, but this really makes the case that this really works. We still have a long way to go, but given this sort of funny business that comes from studying organisms like this, how do we go about the process of organizing NIH research in a way that actually moves in a direction that we want to go? Right? It's fine to understand things in an African clawed toad, but that's not the end point. Right? But if we don't do those studies in the African clawed toad, we're not going to understand what we do care about. Right? It's a tricky business. How do we get there? What does the NIH do? So the NIH has created a research portfolio that has a foundation of that basic research, the fundamental discovery stuff. About 60% of its effort is dedicated to that. And this pyramid has been with this distribution actually for a long time, several decades. In 1945, Vannevar Bush wrote a really critical report called Science to the Endless Frontier, in which he said, basic scientific research provides scientific capital, the stuff that makes the translational research, the linking the basic findings to stuff in the clinic or stuff in social behavioral studies, that then goes out and drives the clinical studies. And we must maintain that level of research. Well, who's going to do that research? Who's going to do that research? And so President Obama had it right when he said an investigation might not pay off for a year, a decade, or at all. That's the way basic research is. And when it does, the rewards are enjoyed by those who bore its costs, but also by those who did not. That's why the private sector underinvests in basic science and why the public sector must invest in this kind of research. So it's the federal government that drives this critical part of the pyramid. It's not that there's not great research, translational and clinical research going on under NIH support. There is. $30 billion is a lot of money, so 15% of that is a lot of money. But the foundational stuff that's not going to get done without the federal support is really carried out by NIH and other agencies that do life sciences research under federal support. So when you think about prioritization, then, knowing what you now know about the complexities of biology and the necessity that disease is similarly complex, you can see that it's not going to work by simply making a list that says, let's make sure that we put a lot of money into pancreatic cancer because it hasn't really moved forward for 30 years, which it hasn't, right? But maybe that's not the way to the end point for pancreatic cancer. Maybe it's going to be through the African clawed toad. So making those priority decisions becomes really tricky. And so in a way, the prioritization is this. I'll drill down on this further as we go, but in a way it's this. Collect lots of data about lots of processes and lots of organisms and use all that stuff to understand and treat disease. Well, that's certainly the priority. It doesn't really tell you much about how you do the details, and we're going to talk about that, or how you make the decisions, and we're going to talk about that. But in fact, as a statement of priority, I think that's not so bad. On January 30th, President Obama rolled out in the East Room of the White House his precision medicine initiative. Something called precision medicine gives us one of the greatest opportunities for new medical breakthroughs that we have ever seen. He had mentioned in the State of the Union talk, he rolled it out at the White House at the end of January, and there's already been a meeting at NIH to talk about exactly how the initial aliquot of funding that was put into the president's budget would be utilized. And the study will go on through the course of 2015 to make some decisions about how that's going to actually work. Well, what the heck is precision medicine, and how does that play into these decision-making processes and prioritization? Precision medicine is essentially Google Maps for biology and disease. You know the way the Google Maps work, and that is that it stacks one layer of information on top of another about any kind of set of parameters that you want to study, and those layers are oriented by geography. So if you were to drop a plumb line through any spot on one of these planes, you would immediately be able to correlate the information about transportation and land use and census tracts and structures and postal codes and so forth. Precision medicine does the same thing, but with a catch. Precision medicine says, let's find all the different kinds of research information that's being collected, epidemiological studies, your individual medical record, your human genome, other omes that you might have heard about, the proteome, the exposome, all sorts of good ohms. The catch is that after you stack these things one on top of the other, dropping a plumb line to find the correlations between them doesn't work. We don't have a common orientation point like geography. And so instead, you have to do that pesky research stuff and find out how the different bits of information are linked. So precision medicine says, gather lots of data and find correlations between the layers of information that we haven't been talking to, that haven't been communicating with each other. And the correlations that emerge from that will inform us more deeply about mechanisms of disease and mechanisms of biological processes than anything we've had before by focusing one layer at a time. So precision medicine then says build a massive data network to aggregate and analyze information from patient cohorts, healthy populations, experimental organisms, to determine disease mechanisms and mechanisms and networks, and that precise health advice, diagnosis, and treatment for each individual will emerge. So instead of statistical groups and risk factors going from 1% to 4%, now you have advice that's specific, drilled down all the way to the level of the individual. So then we can now we can dive a little deeper and talk about the challenges and outcomes of NIH research in terms of prioritization. And so maybe now we can carve out three things. One would be to study all biological processes where they can best be understood. So capitalize upon what biology has given us and being able to look at simple organisms where we can do experiments, all the way to complex organisms where we can't do experiments on people, we just got to study them. Right? And then aggregate, integrate, analyze the vast collections of biological data, creating a knowledge network. And that knowledge network will then be used to inform diagnosis and treatment decisions for each individual. It'll empower further research because new correlations will be revealed that will be the stuff of making new hypotheses for new experiments. And it'll advance clinical care and inform patients and well people who aren't sick and would like to just keep it that way, if you don't mind. This is a tall order, right? But it's a way to drill down on how we can think about prioritization And even though it'll be hard to accomplish, as the president pointed out, it's worth it. It's worth it because the outcomes would be so profound. We would have a research ecosystem that for the first time makes full use of all of the discoveries in basic clinical and social behavioral sciences. That hasn't been done. We talk a good line about how integrated these areas of research are, but the silo walls are still there. And we're not really using all the information on the table. It's a long time before basic research discoveries in labs like mine make it into changing of clinical care and standard of care in the clinic, even though the UCSF hospitals are right next to our research buildings. So that would be fantastic to be able to use the information. Health and health care will be tailored precisely to each individual, as we've talked about. This will result in greater worker productivity, higher quality of life. And then the last one's pretty important. Reduced healthcare costs. Reduced healthcare costs. We are in an unsustainable still, despite the fact that the curve has been bent by the ACA. We are still in an unsustainable trajectory with respect to healthcare costs in this country. And so we can reduce those healthcare costs due to improved prevention and early diagnosis. You probably are all aware that if you can catch diseases early, you can cure them. If you don't catch them early, sometimes you can't. Better control of chronic disease and avoidance of unnecessary tests and ineffective therapies. So when you hear about a blockbuster drug, that's not actually a drug that works on everyone. It's a drug that doesn't kill anyone. And so then everyone can buy it. And it helps some of those people and doesn't do anything for others. Right? You go into a fancy tertiary medical center like UCSF, and they're likely to throw the book of clinical tests at you. Right? Some of them will make a difference for you, and some of them won't. But because we don't know enough about mechanisms, that's what you get. So we think this will drive healthcare costs down. Pretty substantial benefit. So Howard mentioned the way the NIH works. So with those goals then, the challenge is, what kind of organizational structure do you put together, right, that will capture all this stuff? I mean, it's fine to say we should study a lot of different organisms and a lot of different processes. But what structure do you put together so that things move in a direction? And how do you decide who should carry out that work? Right. So there are these topical institutes that fund research. These are the people. Those are the groups that actually have the budget to come to Congress every year and get a budget to an exact dollar amount to spend in their areas of focus. And there's the Center for Scientific Review, which is separate from all these. It's not beholden to any of them. It operates independently within the NIH to evaluate the grant applications that come in. Every NIH grant, the R1 grant is just NIH jargon for an investigator-initiated research grant. The investigator sits down at her computer, has an idea that she's curious about, writes out a grant on any topic and sends it in. So each one of those grants, and there's 80,000 of them a year or so that come into NIH, are actually reviewed twice. The first level is by a study section. This is the peer review thing that we're going to talk about. So it's a group of expert scientists in an area of study, pros that have achieved some notable contribution in an area, and have been asked by the NIH to volunteer to serve as peer reviewers of grant applications that are coming in their field. So their stock in trade, their currency is knowledge, and their role is to assess the scientific merit of the applications. The second level of review is carried out by a group of scientists and non-scientists that have been pulled together by those different funding institutes. and their currency is currency. Their job is to assess the relevance to the institute and program portfolio of the grant applications that come forward to them from the study sections, and say, this got a great review, do we care about it here in the National Cancer Institute, and if so, we'll give it some money. So one of the geniuses of the NIH system is that it actually uncouples the decision about merit from the decision about money. So the people that are making the merit decision don't have to worry about whether there's enough money to do this. They simply say, if there was an infinite amount of money, is this a great study? Right? And if there's an infinite amount of money, is this still terrible? We should get rid of it. Right? So what is NIH peer review then? And how does it work? This is an interesting process because it is an evaluation by scientists of the scientific merit of proposed science that comes to Bethesda to be funded. 83% of that $30 billion is allocated by this process. Now, if you were to pick up the Post and find that a special interest group had somehow managed to get control of $30 billion of federal money and spend it on themselves, you think we got a scandal here. Well, that's NIH peer review. It's working scientists, right? I have an NIH grant. I serve at the NIH to do peer review. It's working scientists who have their own skin in the game for that very money that are making this assessment and funding or not funding their friends, enemies, and people they don't know. We'll talk a little bit more about that, but it's an interesting way to think about the process. The process is managed by the Center for Scientific Review, independent of the funding institutes, and the reviews are executed by these committees, study sections, committees of experts in an area of study, right, that are made up of working scientists who volunteered to come to do this work. Now, the people who serve on these study sections, when we're saying volunteer, we're not kidding. To get ready for one of these there's 3 meetings a year. To get ready for one of those meetings takes essentially 2 weeks of solid work, reading these grant applications and making an assessment of their quality. So a member of a study section who signs on for a 4-year term is saying, okay, I've got a lot of things to do. I've got a family. I've got my own research laboratory. I've been working at my university. But okay, I'll give up 6 weeks a year for 4 years in order to serve the community in this way. So people take it pretty seriously. And they come to Washington and they have to buy their own coffee and buy their own lunch. It's really pleasant. Really an honor. But in fact, it is an honor, and the people who do it take it very seriously, and the process works incredibly well. What do they do? Well, each reviewer has to address a set of these 5 criteria. They're pretty obvious when you look at them, but it was the subject of a 2-year fight in the 90s. I took a major part in to think about the impact of the work. What difference would it make if this work was done? Would it move the field forward? Even if the experiments were perfect, if it doesn't move the field, then we're not interested. Right? Is the approach right? Is it innovative? Is the investigator well qualified? Is she working in a place, in an institution, where the environment will actually help the work go forward? So every reviewer has to comment and actually give a little preliminary score on each of those 5 criteria. And then give a score, 1 through 9. You can see the descriptors to kind of span the scores out. And those scores are then entered by every member of the study section in the room. by integer scores, and then this overall impact or priority score is computed as an average of all the committee member scores, and happen to be multiplied by 10, so the scores go from 10 to 90. 81 gradations is a lot. It's probably a lot more than the human brain can actually process, but it's better than the 501 that were there before this change was made. So complexities of peer review and the limits of knowledge. Does the process really work, right? Are there things wrong with it or problems? Where do we need to go in context of the way that work is going? This first point is, in a way, the most important in terms of thinking about whether the process gets rigged in some way. And that is that it is remarkable how effective the processes are that the NIH uses to acknowledge the intrinsic conflict that comes with peer review. There's no getting away from it. There's no denying it. There's no saying we're going to make policies that will make it go away. It's working scientists that are making these judgments, that are making decisions, recommendations about what should happen to the funding of people that are working in their own field that are trying to tap the same money that those working scientists are trying to tap. So the conflict is there, but the management of those conflicts is extremely good, and people pay, as I said, are very serious about the process, and it works well. It has to operate on a massive scale across a broad scope that we talked about. So that's challenging to make that work. So there's a lot of study sections, so 250 of them or so, areas of study. Incorporates these complex metrics for merit and success. It's not perfect, and there is a Center for Scientific Review Advisory Committee that is at work constantly, 2 meetings a year, to evolve and improve the process. It's able to work in ways that will support different types and styles of research: basic, translational, clinical, social, behavioral, and research that both moves things forward in a relatively linear way, and research that is transformative, that will make big quantum leaps in our understanding because of some brilliant insight or new technology. Is NIH peer review the best system? Are there other ways to do it? NIH grants are bottom-up. Investigators initiate proposals, and they're then evaluated by fellow scientists. The alternatives, here's 3. There's probably others. Central planning of research topics and projects. Hierarchical control of the process by the guys who've been there the longest. Or just spread the money thinly over the country. By any measure, the NIH peer review system is not the best good way to allocate resources for biomedical research. It's the only good way. Other countries are always visiting to try to emulate this system because they see that it works the best. By any measure, NIH peer review is best, whether it's in scientific publications, the new amazing discoveries that come out in both concepts and technology, account of the Nobel Prizes, since NIH research started in this system in 1946, or the effectiveness and efficiency, something I call the Starbucks test that I'll show you in a minute. So the ongoing commitment to excellence and enhancement offers continued promise for extraordinary scientific discovery and development. However, the NIH budget has been stagnant since 2003. It's been essentially flat or declining. The spending power of the budget has gone down by a quarter. Only 1 in 7 meritorious grants, only 1 in 7 meritorious grants are actually funded because of the current funding situation. Right? So that's pretty tough on the scientists who are trying to make this their career. And they get a meritorious score, but only 1 in 7 of them gets funded. This against the fact that there are enormous opportunities and advances that can be had and that we're losing because of the lack of resources. What's the Starbucks test? It's a test for effectiveness and efficiency of, I made this up, so. For effectiveness and efficiency of NIH research. And so here's a version of it. Ready? Over the past 40 years, cardiovascular disease mortality has declined by 60% in this country. Right? So cardiovascular disease cured? Nope. A lot of people still dying? Yep. But 60%. This translates into 1 million fewer deaths per year. So here's the test. What was the NIH investment per American citizen per year in $5 Starbucks beverage units that saved 1 million lives a year? Any thoughts? 1. $5 a year. Would you give up 1 Starbucks per year to save 1 million lives? Just think what we could do if you gave up 2. So, as Howard said, NIH has been called the crown jewel of federal spending. I don't know if it's a crown jewel. Hopefully we don't have a crown. But in terms of quality, efficiency, and impact of the way that the federal dollar is spent, the NIH does a great job. Whoops. Oh, boy. So it's really thanks to that, the peer review system, the volunteer efforts of working scientists who come to do this work of making this assessment, and of the federal legislators who make decisions to allocate funds to the NIH that makes the NIH the crown jewel. So what you can do, and an important thing that Howard mentioned is that that money, that $30 billion, is not being spent in Bethesda. Some of it is, 10%. But most of it, that's being evaluated by extramural scientists from your districts and your states, is going to your districts and your states. So making those decisions about the way that that money is spent is really the key thing. And your opportunity is to be able to provide those funds to recognize that this is a really efficient way to really have an impact on American health. And in the course of doing it, really feed an endeavor that not only enriches kind of the intellectual life of our society, but also has a great economic impact. So let me stop there and see if there's any questions you might have. I'd be happy to talk about it.